All right, good morning. Back. Okay, and remember we've got an exam number 3. It's Friday, 2 o'clock. We just got room assignments, gonna get some better rooms. Not that we succeeded. So we'll be sending out those room assignments this afternoon. I'll do a Q&A session at 11:45 to 12:45 today. Should we do like a cram session as well, extra bonus Q&A session today? Would anybody like that or not? I can do that on Thursday. Okay, I'll probably Thursday afternoon, I'll put it up. And you can feel free to send questions in ahead of time, like email me, and I can, I'll either email you a direct response. I can also address those questions. We have enough time in Q&As in the Zoom Q&A. So I'll post that Q&A. Those Q&As are, of course, recorded with transcripts and everything, so you can review that. Any questions on content? Content is going to be cardiovascular system, basically all of the lectures that we've already done, but not including today. So cardiovascular system, respiratory system, immune system, fluid and electrolyte balance, also known as the kidney. We also have posted a practice exam, which is 2015's exam, and my own little draft of a review packet for this exam, as well as Professor Bearcat's very thorough review packet, which I think is probably relevant. Most of the stuff is relevant. If it's not in my lecture, then it's probably not relevant. But if it was, always good to get another sort of perspective on knowledge. Any other questions? Cool. All right. So let's get started. Okay. Get rid of this thing. How do I get rid of this thing? Go over there. Okay. And let's shrink that. Okay. Back to this guy. Anybody know who this guy is? Arnold Schwarzenegger, former governor of California, times for Olympia, right? Star of many movies, most of which were before your time. Terminator, Terminator 2, all of those ones, right? So, one sort of hallmark of this absolute unit was that he had why can't I shrink this? Okay. Basically, his, physique was sort of unique for the time, right? And it didn't just happen just by lifting weights. It was really developed through an interaction of the endocrine system. Signaling molecules like testosterone, growth hormone, insulin, were all sort of collaborating together, sending messages to each other in order to tell the body to take up glucose, turn it into the muscle, right? Turn the muscle into these particular targeted locations. It's like a group chat between all these different components of its body. So how was that actually achieved, right? And how did he turn those signaling molecules into the growth? Would that signaling molecules, do you think they would be a catabolic signaling molecule or anabolic signaling molecules? Catabolism is the breakdown of body tissues and turning it into energy. Well, anabolism is the construction of tissues from raw materials. So anabolic steroids, not only the ones that were naturally within his body, being sort of triggered through the release by, deadlifts or squats or whatever else he was doing, but also the leveraging of knowledge and scientific knowledge of the endocrine system allowed him to use synthetic anabolic steroids that would build him up to this particular level. Now, of course, those have been, more and more abused in the bodybuilding industry until now. You have these incredible caricatures of people in a very unhealthy state that are abusing anabolic steroids. But just to be clear, endocrine system has to illustrate this though, that the endocrine system has incredibly powerful effects on the body, both on composition of the body and also on behavior and many other aspects from stress to metabolism. Okay, so we're gonna sort of learn about all of those different signaling molecules in the next 2 lectures. Okay, so the key concepts for the first lecture that I'd like you to sort of hone in on or focus in on are classifications of the different types of chemical signaling. Okay, not everything is a hormone. There's autocrine signals, paracrine signals, all those things. We want to understand how the signaling molecules are transported to their target locations and how they're going to generate the specificity of signaling so that the same molecule, man, there's that really high-pitched ringing again, right? The same molecule can do different things depending on what target tissue there is. It's attaching to. You're going to want to know every hormone that we discuss, the source of the hormone, how it's sort of synthesized, and what is the originating location of that hormone. Then what is its target? Okay, and what is that functions? What is the function of that hormone? How does it achieve its functions through actions on its target? And then finally, I think by the end of this lecture, we'll probably stop at the function and dysfunction of the pancreas, which is really important for regulating blood glucose levels. Both the alpha and the beta cells of the pancreas can cause release of hormones that will take glucose up, release glucose into the bloodstream, which is one of the most important signaling molecules and one of the most common, and dysfunctions of that are one of the most common forms of disease in America, right? Diabetes mellitus. Okay. All right, so there's two major systems for communication between your body's tissues, okay, that are going to communicate and control to the rest of the tissues. We have the endocrine system, which we're talking about today and this week, right? Which is going to be a slow and sustained system where you're releasing chemicals. They're traveling perhaps through the blood or through neighboring regions at a relatively slow time scale, you know, from the order of seconds to minutes to hours. Okay, and that's going to cause sustained responses throughout and coordinate the responses across a large number of your body's tissues. Now, for particular behaviors, you're going to want to move faster, right? You see some, if you want to stand up and deliver a message, right? Or like I'm doing right now. If I want to talk to somebody, or if I want to run, or if I want to move my body around, you're going to want to coordinate that in a much faster way. So you're going to also have to have a parallel system for communicating between your body's tissues, which is the nervous system. That's going to be conducting electrical signals. Okay, so the nervous system and its relationship to the muscles in the body is really going to be the last 4 lectures of the course. Okay, so we've got endocrine this week, we've got sexual reproduction next week, and then we've got nervous system and muscle systems to finish us out for the last 2 weeks. Okay. So now zooming in on the endocrine system. So it's one of these 2 basic systems for communicating and regulating responses. What does it actually do? Okay, it does a huge number of things, right? Anything that has to coordinate a response across your body's tissues, right, is really going to be signaled through is going to be coordinated through responses of the endocrine system. So you can imagine the development of the body from an embryo all the way up to a fully grown adult is going to be involved in things like growth, hormones like growth hormone and other endocrine signals are going to be orchestrating that sort of transformation. Then we have drives, biological drives to get resources like hunger, right? If you're hungry, if you need to increase the amount of glucose in your body or amino acids, we have very specific hungers and drives for that. Thirst, the ability or the drive to consume water to maintain fluid balance in the body is going to be driven by releases of particular hormones in the endocrine system. Our responses to stress, whether that's acute stress or chronic stress, is going to be regulated by the release of cortisol, which will change your blood glucose levels and allow you to have a higher sort of level of glucose in the body that will allow you to respond to stress. Of course, if that cortisol levels are maintained for long periods of time, then that could, due to stress, that can lead to dysfunctions of the body systems. Mating, sexual reproduction, sexual drives, these are all controlled through the release of endocrine hormones. Energy metabolism, how much of our cells are burning fuels and how much of the cell's job is actually taking in fuels for storage. That's going to totally influence our body's composition. How much fat do we store? How much muscle are we building? How much energy do we have throughout the day? All controlled by things like thyroid hormone. And then finally, many other behaviors, I should say. Okay, basically any sort of intrinsic drive that is motivating you to do something is generally going to be controlled by 1 or 2 or more hormones that are being released in your brain or your various glands, your adrenal gland, gonads. All of that stuff is sort of floating through in this stew of material and acting on brain tissues like the hypothalamus that have specific nuclei within it that would cause you to have particular drives to actually go out and do something. Okay, so what are the different classes of the chemical signals here? And we can really break them down in a couple different ways. One of the ways that we can break them down is just in terms of how far they are, how far the signal has to travel, and what is the method of that transmission. Okay, So the closest signal that you could kind of imagine a cell doing, okay, releasing, is a signal that releases and binds onto receptors on its own cell surface. Okay, that's called an autocrine signal, a self-signaling mechanism. Why might we want to do that? We might want to have some sort of negative control feedback system, maybe within the cell. It says, okay, I'm going to release a certain amount of this molecule, but I don't want to release too much. I don't want to go out of control. Okay, so that I'll have some sort of receptor here that's going to react to binding of that molecule and say, turn down the production of that molecule. Or if I don't have very much activity on this receptor, turn up the production of that molecule. So this is a built-in negative feedback regulation. It's oftentimes an autocrine signal. Then we have paracrine signals. If we go a little bit further, paracrine signals are signals that are released by one cell, and they can diffuse over and travel over to their neighboring cells to act on those cells. So the paracrine signal is next nearby. Is there any recent examples that we've seen of paracrine signaling in the kidney, for example, in the juxtaglomerular apparatus? So we can regulate the overall level of blood flow within to the afferent glomerulus by release of various paracrine signals from the macula densa of the juxtaglomerular apparatus. Okay. Now those paracrine signals, okay, they can be sort of diffusing across cell membranes. They also can be signaling via contact-dependent mechanism where you actually just grab onto a receptor and you're touching it. We can see this all the time in the immune system. Cytotoxic T cells will grab onto those peptides and things displayed on NMHC class 1. And through those contact-dependent mechanisms, release particular molecules onto it. So that's a form of paracrine signaling. Now, the paracrine signals are noteworthy in a sense, are distinct from endocrine signals, also known as hormones, in that they don't have to be released into the blood for them to act on their targets. Endocrine signal or hormone signal is where you're going to get a release into the bloodstream. Now, what is that going to do? That is going to allow the signal not to be confined to its actions locally, but actually be able to travel throughout the entire body. Okay, so it's going to circulate through the body, and wherever it finds a receptor that's specific to that particular molecule that's released, you're going to get an endocrine signal event occur. So the hormone is going to go everywhere, but it's not necessarily going to act on every body tissue, right? It has to have the appropriate receptor for that. There's some other specialized forms here that we can talk about. Gap junctions, right? These are, I wouldn't really call it it's not really a hormone or a signaling molecule, okay, but it is a way for cells to communicate between one another, right? They have these pores that are basically little direct routes between the cytosol of one cell to the next cell to pass small molecules and ions. You can pass electrical current through cells like that. Another form here is synaptic transmission, which we'll go into quite a bit of detail during the nervous system. So this is a hallmark of how information is transferred within the nervous system between neurons. So we'll have basically a cell body here, the soma, that will produce, that's going to have some long elaborate processes, maybe it's called an axon. It'll have little vesicles of our chemical here. If it's a chemical that's going to be acting within the nervous system specifically, it'll be called a neurotransmitter. It'll be released. So the presynaptic term on the pre side of what is known as a synapse, which is a small gap between two cells. Okay. And then we have a postsynaptic cell that will have some receptor or suite of receptors that is able to bind the signal and then transmit signals to the postsynaptic cell. And that could be through opening of ion channels, causing electrical currents to flow, or it could be through binding to G protein coupled receptors, driving secondary messengers that'll have a longer sustained effect within that postsynaptic cell. So that whole process is called synaptic transmission. There's also a version of that where we have what's known as a neurohormone. where one of these neurons will produce a particular compound that, rather than being released onto a neighboring cell, will be released into a capillary bed. And then that neurohormone that's produced in a neuron, but acting as a hormone, will travel throughout the body and act on target tissues. And finally, we have pheromones, which is where hormones released from or molecules released from a cell can act on other individuals of the same species. Now, you see this all the time in some mammals like, rodents or dogs. They're constantly sniffing and they have a very robust chemical communication. If anybody's ever owned a dog and they walk the dog, all the dog wants to do is go to sniff the pee. And then they want to go pee on the pee because they're just talking to their friends, okay, around the neighborhood through their pheromone-based signals. Okay. And these signals can be really powerful. Okay, ants. Have you ever seen videos of ants doing really complicated things like cutting up leaves and like building elaborate structures out of them? All of that is done through pheromone signaling and the collective action of groups. Now in humans, there seems to have been a strong sort of environmental or evolutionary sort of suppression of pheromone signaling. The organ called the vomeronasal nasal organ, which is used for signaling in other mammals, doesn't seem to be present in humans, but there still may be some ways for pheromones to signal in humans. It's still, the research is a little bit unclear. Certainly it's a part of marketing for things like perfumes. So we're going to drive some particular behavior through pheromones by wearing this perfume or something. But exactly how that might mechanistically work is unclear. Okay. So that's like classification of these chemical signals by distance and method, but we can also classify them by how they're composed. Okay, that is, what is their structure? How does their structure influence their signaling and their functions? There's three main classes of these structures, okay? We've got polypeptides, we've got amines, and we've got steroids. Okay, so polypeptides. A polypeptide is just a chain of amino acids. It's derived from a protein, usually. So you make, we've got our DNA, we make our RNA, we make our protein, and then we process that, which is just a chain of amino acids. We've sort of translated this mRNA transcript in a chain of amino acids. So that's a peptide by definition, right? And then there's maybe some processing that's going on in the Golgi apparatus that cuts stuff up and turns it into a polypeptide, okay? A protein, a chain of amino acids that's more than one amino acid length. Great example of a polypeptide protein or a polypeptide signaling molecule is insulin. Okay. Classic example here. Then we have amines. Amines are signaling molecules that are from a single amino acid, okay, not a whole chain of them. Okay. And these are usually derivatives of either tyrosine or tryptophan. And depending on whether you're tyrosine or tryptophan, you're going to have a different level of water solubility. Okay. So for example, this amine, epinephrine, is water soluble. This amine, thyroxine, which is basically a thyroid hormone that influences metabolism, is going to be lipid soluble. That's going to influence how you're going to be able to get to your destinations, right? And then finally, we have steroids. Okay. These are derived from cholesterol. Cholesterol is not just some bad thing that's going to clog up your arteries. No, it's an intrinsic component to all of your cell membranes. Okay. Your cell membranes are full of fatty acids and cholesterols. They have these sort of complicated structures. They're really greasy. Okay. And there are biosynthetic pathways that will modify the exact sort of chemical moieties on the edges of these things that will completely change their signaling characteristics or which receptors they can bind to. So many of our sex hormones, regulation of glucose levels are done through, and stress levels are done through steroid hormones. Okay. So we're also can split these into the water soluble and lipid soluble. So you can imagine that these are going to have to take two different pathways to get to do signaling inside the cell. That's going to be influenced by whether or not they are dissolvable within the blood or have to be carried somewhere within the blood, have to be carried within the blood, and how they're going to actually be able to pass into a cell, right? If you're water soluble, you can't get through a cell membrane. If you're lipid soluble, you can diffuse right through. Okay, let's look a little bit more on insulin here. Okay, so insulin is our sort of classic polypeptide signaling molecule, and it is a hormone that is produced in the beta cells of the pancreas. It's involved in regulation of blood glucose levels. It causes blood glucose to get taken up into the cells in the body. And within these beta cells of the pancreas, the initial form, the initial translation into a protein happens in the rough ER, and it creates this thing called pre-proinsulin. Okay, it consists of a signaling sequence, this sort of long chain here that goes all the way to here. That then gets processed. Okay, the signaling sequence gets cleaved off. Okay, in the ER, it gets turned into proinsulin, where you have disulfide bonds starting to form and causing a change in the structure of this insulin. And finally, in the Golgi apparatus, this gets cleaved off into a bound A chain and B chain, okay, as well as what's known as a C peptide. Okay, so this floats, these things are all sort of stuck together, and they are going to get co-released by the beta cells. Okay. Now, this part is insulin. This is the only thing that actually does something within the body. Okay, travels around, finds the insulin receptors, increases the blood glucose levels, or decreases blood glucose levels, causes glucose to go up inside the cells themselves. But what about this C peptide? This doesn't do anything, okay? But it does get released, and it is useful because this is what we actually measure in tests of whether or not your beta cells are producing insulin. Because it gets co-released, because it's just floats around instead of it gets bound to stuff, you can actually use that to measure how much insulin is getting released by beta cells. Okay. Just for testing for these C peptides. Okay, so that's sort of the structure of insulin, right? We basically have this chain of amino acids here, this polypeptide chain, gets cleaved, gets bound by disulfide bridges, and that becomes the active form. And what about some examples of amines? So amines are going to be produced in a variety of different areas. You can produce it in the brain, the thyroid gland, and the adrenal gland, and the gut. That does like serotonin. The brain does norepinephrine, dopamine, things like that. Here is sort of just an example. You do not need to know all of these different like structures here. I do not expect you to know a chemical structure of any of these things. I could not draw it for you myself. Okay. And you don't need to know like all these different conversion enzymes. This is not organic chemistry. You get to learn maybe this, how to do these syntheses in OChem. But what is important here to understand is that a single amino acid here, L-tyrosine, can be transformed through enzymatic steps into a variety of different neuromodulators or different neurohormones that cause dramatically different effects within the body. Okay, so L-tyrosine turns into what's known as L-dopa. This is a precursor to dopamine. L-dopa is actually, that gets turned into dopamine. Dopamine is something within the brain, which is going to drive sort of motivational state, right? And signal rewards. Okay, dopamine gets transformed into norepinephrine. This is going to also be involved in overall general arousal levels. And norepinephrine then gets turned into epinephrine, which is basically adrenaline, which can really change heart rate levels, change what areas of the blood vessels are vasoconstricting versus vasodilating, and is the key molecule for driving a fight or flight response. Okay, so this same originating amine here, tyrosine, gets transformed into these three different signaling molecules. And these are all water soluble. So if you're coming from tyrosine, you're the water soluble ones. Okay, finally, we've got our steroids. Okay, so these are going to be produced in the adrenal glands and your gonads. Gonads are testes and ovaries. And they are all derived from cholesterol. Once again, you don't need to know any of these steps. Okay, but what you do need to know is that they same originating molecule here can get transformed into all of these different other signaling molecules that have very distinct functions and even different classes of functions. Okay, so we have all these colored ones. We have progesterone, which is sort of a precursor to many of the sex hormones and is involved in the menstrual cycle. We have the ability to conceive, ability to become pregnant. These can get transformed into corticosterone, aldosterone, which is going to be involved, of course, in the regulation of mineral levels within the blood. We have cortisol, which is involved in, which is basically the major glucosteroid of the body, which is going to regulate blood glucose levels along with insulin, sort of counteracts the impact of insulin. Then we have these sex hormones, okay? Both sort of female and male sex hormones are things that produce, have male, promote male production of male specific characteristics or female specific characteristics. So estrogen and estradiol being the female specific ones and testosterone being male specific. So all of these hormones, okay, are going to be produced either in the adrenal gland or gonads and act on diverse tissues to achieve a variety of different goals. Okay, now let's take a look with a little bit more specificity now into how the signaling of a water-soluble hormone might differ from a lipid-soluble hormone. So let's just start with any sort of secretory cell up here. Okay. From the very beginning, how that hormone is going to be released is going to be different based on whether or not you're water-soluble or not. Okay. If you're water-soluble, you're going to be sort of packed into these little vesicles. Here are these little balls, okay? Because you're going to be dissolved in some sort of fluid, okay? They're going to have a fusion of this vesicle with the plasma membrane and a release of that water-soluble hormone out into the interstitial fluid that can then quickly travel into the blood vessel through gaps in the endothelial lining, okay? And that will then be transported throughout the blood, okay? In a lipid-soluble hormone, you're actually going to be released through the plasma membrane itself, okay? Because you're actually going to be, oftentimes, these will actually be produced from the cleavage of elements of the plasma membrane. Cholesterols or fatty acids can enzymes that act within the plasma membrane itself that are going to convert or cut up different components of that plasma membrane and turn them actually into signaling molecules. From there, they have to be transported through the blood by some sort of carrier protein. The major carrier protein that is actually the most abundant protein within the blood that is dissolved in the blood is albumin. Okay, this is going to bind on, this is basically a generalized binding protein that can bind onto many different hydrophobic proteins. Okay. Now, to sort of complement that albumin, we also have other specific globulins for specific signaling molecules. We have a glucocortical specific globulin for binding cortisol. We have sex hormone specific globulins for binding testosterone, estrogen. Okay. And what we mean by globulin, we've heard of globulin before, globulins. These are just basically any globulin is like a protein that is sort of has some sort of spherical shape and can be dissolved within the blood. It travels throughout the bloodstream. That's what a globulin is, a protein that travels throughout the bloodstream. And it can bind on to other proteins. So immunoglobulin is going to bind onto antigens, right? Sex hormone globulin is going to bind onto sex hormones and transport them to target tissues. Okay, so when we get to our target cell, our water-soluble proteins have to bind onto, can't get in, can bind onto the cell surface. A lipid-soluble hormone can actually diffuse into the plasma membrane. From there, they can act to either have a cytoplasmic response or regulate gene regulation through second messengers, or bind to receptor proteins in this case, and then translate to the nucleus and drive direct transcription. So we can sort of look at this a different way, okay, which is maybe a less pretty way, but actually maybe in some ways more informative. Okay, we've got our hydrophilic hormones, so peptides, many kinds of peptides and proteins and amines Lipophilic hormones, which are mostly the steroids and the thyroid hormones. Okay, thyroid hormone is derived from an amine, but it is hydrophobic. Okay, so a hydrophilic hormone travels around, hits a plasma membrane receptor. It cannot get in. Okay, it binds the plasma membrane receptor. Often these are G protein coupled receptors. They're going to cause the production of some second messenger. Okay, might be cyclic AMP. Okay, and that is then going to turn on various protein kinases, which will phosphorylate things and drive the intracellular response. On the other hand, you have lipid hormones or lipophilic hormones, okay, that are going to be sort of in equilibrium with their carrier protein. So they're always floating around, they're binding on the carrier protein to get transported. Sometimes they fall off. If they fall off adjacent to a plasma membrane, they can just slip right into the plasma membrane. Okay. Now, within there, okay, we can either sort of bind onto a plasma membrane receptor that then drives a similar pathway as these hydrophilic hormones, okay, drive second messengers, or the more commonly expressed signaling pathway is once this lipophilic hormone gets into the plasma membrane, okay, it is going to run into a cytoplasmic receptor. So this is a receptor that is soluble within the cytoplasm, but has a sticky sort of oily part that can stick onto this molecule, maybe it's estrogen, okay, just as an example, bind onto it and then pull it out of the plasma membrane, and then float around and get transported over into the nucleus. When it reaches the nucleus, this can then bind onto specific locations of the DNA, promoter regions or enhancer regions, and act as a transcription factor. That will either promote or suppress the transcription of mRNA from DNA. And of course, if you're transcribing mRNA from DNA, that is then going to get translated into protein. So these hormones, once they're bound to the cytoplasmic receptors, actually directly act on the DNA itself to drive direct changes in protein production. Any questions on this stuff? Okay, so let's look at a specific example. Let's look at epinephrine. So here is our lipid insoluble, doesn't like lipids, hydrophilic, loves to float around in water or in your interstitial fluid or in your blood. Doesn't need a carrier protein. Okay, it travels around. Okay, gets released. Okay, it travels around. Epinephrine, also known as adrenaline. Okay, it binds onto our G protein coupled receptor here. There's a transformational change. There's a conformational change in that G protein coupled receptor. It exposes some little part of this G protein that causes the G protein that's normally associated with this G protein coupled receptor to unbind from it. Okay, it's going to travel over through the plasma membrane here and grab onto adenylyl cyclase, okay? And that is going to then cause adenylyl cyclase to start catalyzing the conversion of ATP that's floating around into cyclic AMP. So this is basically taking off 2-phosphate from the ATP, okay, and making sort of a circle out of what's left. And then the cyclic AMP is then going to travel over and attached to protein kinase A. It's going to activate protein kinase A by relieving some regulatory subunits. And then this guy is going to run around and start phosphorylating all sorts of crap. Okay. It's going to, and that ultimate end goal of this sort of chain reaction of events from what it phosphorylates is going to inhibit the synthesis of glycogen and promote the breakdown of glycogen. So does anybody know what glycogen is? Have you heard of what glycogen is yet? Yeah, okay, right. Glycogen is basically a chain of glucoses that are stuck together. Okay. You can, if you have a bunch of glucose in your blood and you want to store that glucose for later, right? Maybe you want to have some energy, you want to have glycogen taken up into your muscles. Okay. The muscles in your body can store 300 or 400 grams of glycogen within the muscles themselves. And then when you have low blood glucose levels, or when you're exercising, you need to use those muscles. Your body can break down the glycogen into glucose, okay, right there in the muscles themselves. And that can provide an energy substrate for exercise and use of those muscles. Okay, so epinephrine, What is it involved in? Fight or flight response. Release of epinephrine does fight or flight. What is it going to do? It's going to cause a promotion of the breakdown of glycogen, so an increase in the availability of glucose within your muscles and within your bloodstream. It's also going to inhibit the synthesis of picking up. You can see why that might be very useful for a flight or flight response, right? We want to have more energy available to our muscles in order to escape. Okay, now let's do one example of a different, of a lipophilic one. Okay, estradiol, which is major female sex hormone. It's going to interpolate into the plasma membrane. It's going to bind to this estradiol receptor. Okay, the estradiol receptor bound to this hormone complex is going to translocate to the nucleus of the cell. Okay. What can it do in the nucleus of the cell? Well, it can actually act as a transcription factor and turn on the expression of a large variety of genes. It's not just one gene, okay? But in this particular example here, I mean, it is going, so estradiol is basically a growth hormone for female reproductive tissues. In this case, it's going to be activating the transcription of vitellogenin, okay, which is a gene that is a protein that produces a protein that is a component of egg yolks. Okay, so you're gonna create this protein. It'll get transported by the blood into the ovary. The ovary will then incorporate that into the yolk of eggs. Okay. So just an example there. Now let's look back. Let's look back at that epinephrine, because I think epinephrine is a great example of this concept where a single hormone can have distinct effects dependent on the receptor that it binds to, okay? And how that receptor is coupled to distinct intracellular pathways dependent on what cell or class of cells that receptor is expressed in. So epinephrine, okay. The overall thing that epinephrine is doing, right, is driving fight or flight response. But we might want to, increase the blood availability to our muscles in the fight or flight response while simultaneously decreasing the availability of blood to our gut where we want to digest things. Okay, we don't have time to digest right now. Okay, so we want to have a vasodilation in one area, but if we vasodilate one area, we would have a drop in blood pressure. So we want to, unless we have a complementary vasoconstriction in a different area. So we want to maintain overall blood pressure. We're going to vasodilate muscle. We're going to vasoconstrict the gut. So how are we going to actually achieve that? Well, epinephrine is released, okay, by the adrenal cortex. This adrenal gland on top of the kidneys gets into the bloodstream. It goes around, quickly hits these alpha receptors, epinephrine alpha receptors, in the intestinal blood vessels. That's going to cause a second messenger cascade that causes vasoconstriction. And that's because this alpha receptor interacts with a different G protein than the beta receptor. So the G protein that the alpha receptor interacts with is going to cause one type of second messenger cascade to occur. The G protein that's coupled to the beta receptor has a different cascade that gets activated. Okay, that's going to then cause different differential effects in the target tissue, even though these are all vascular blood vessels, right? One's expressing alpha receptors, one's expressing beta receptors for epinephrine. So we'll get dilation in this case, constriction in this case. Now, we also, okay, can see that, so this is basically the same type of tissues, but are expressing different classes of the receptor. And then we can have the same receptor, but expressed in different tissues. Okay. So here we have a beta receptor that's expressed in the blood vessels. Okay. If we express that same beta receptor in liver cells, that is where we are going to have a breakdown of glycogen and a release of glucose through that second messenger sequence. So different intracellular proteins are going to give rise to different cellular responses because we're in two different cell types here. Yeah. why it goes into by what goes in the leg. The shot of epinephrine, man, didn't you see Pulp Fiction? They do it right in the heart, right there. You haven't seen, did anybody see that movie? Early Tarantino, right? So, yeah, so epinephrine can go anywhere. As long as it gets to the blood, it'll be effective, I think. But you probably want to get it into a place that is going to be, it really doesn't I think it doesn't actually matter. I think in the case of like Pulp Fiction, they did it for effect, right? But if you're doing it for, epinephrine is also useful for, allergic reactions. So if you have swelling of the throat, right, vasodilation of the throat, you can't breathe anymore, you're getting an anaphylactic shock. There you go. So an EpiPen, stabbing yourself with an EpiPen will inject epinephrine into your bloodstream, will then cause vasoconstriction of those of certain tissues and elevate heart rate, induce a fight or flight response. Okay. Other questions on this stuff? Okay, so here is the list. The master list of pretty much all the hormones you know. What I want you to know is origin, target, and function. Okay, name, origin, target, function. So we have a variety of different endocrine glands here. We've got the pineal gland up here, sort of sitting in the sort of the core of the brain. This is going to be involved in regulating your circadian rhythms, okay, with regulating your sleep cycle and how genes sort of change their overall expression levels throughout the course of a day. Then we've got the hypothalamus that is sort of below the thalamus, which is sitting right here. And that is what I've mentioned has all these different nuclei that control these very strong behavioral drives. There are many signaling hormones that are going to drive activity within the hypothalamus, some of which are not even known. Where do you think, feeding behavior is a great example. These GLP-1 agonists that you might have heard about, Ozempic and things, which can really influence the amount of drive for food that people have, that is going to be acting through actions on the hypothalamus. Then we have the pituitary gland, which is really split into two different structures, anterior and posterior pituitary. The anterior pituitary is a huge number of different hormones that are going to regulate everything from milk production to overall body growth. Posterior pituitary is more like interactions with sexual reproduction. Then we have thyroid gland. This is right here in your neck, okay? And this is going to produce thyroid hormone that regulates metabolism. Parathyroid gland is going to be actually embedded within the thyroid hormone that regulates calcium levels in your blood. Then we have our adrenal glands. These are the things that sit on top of your kidney. They're going to release, we've got epinephrine and norepinephrine that we just talked about, so fight or flight response stuff, constriction of blood vessels, increasing metabolic activities. We've got glucocorticoids and mineralocorticoids in the outer sort of rind, the cortex of the adrenal gland, and that's going to regulate blood glucose levels and mineral levels like potassium. And we've got the pancreas. This is all involved in regulation of glucose levels, blood glucose levels through the alpha and beta cells through insulin, glucagon. We've got the ovaries and testes, the gonads, which are going to be all of our sex hormones and a lot of anabolic steroid production. Okay, so that's sort of a general overview, and we're going to look at each one of these in excruciating detail in the next, lecture and a half. But before we do the hormones themselves, let's cover a few other signaling molecules to just get them out of the way. Okay, 1, nitric oxide. Nitric oxide, this is a gas that can be produced by the body, NO, not nitrous oxide that you get in the dentist office or on TikTok. No, this is nitric oxide. This is going to cause vasodilation of tissues, okay? We have a nerve signal that comes in here. It's going to activate the endothelial lining of these blood vessels, okay? That's going to produce nitric oxide. This is going to diffuse. It's a small gas. It can diffuse right through lipid membranes. It can also diffuse in cytosol. It can just pass right through. It flows over here. So short distance away, diffusion is then going to act on the smooth muscle of these blood vessels and cause vasodilation. Okay. So if we have a level of oxygen, what is going to signal this? Well, if we have a drop in oxygen levels in the blood, then the brain is going to drive this electrical signal that's going to cause NO production, cause vasodilation. That will then increase blood flow to those tissues. Nitric oxide production is a target of many of Cialis, Viagra, all of those things that increase blood flow. That is going to act through nitric oxide. Talk about that in a few lectures. Then we've got, okay, so this would be like a paracrine signal, right? A local signal. Another class of paracrine signal, very important class of paracrine signals, are the prostaglandins, named because they were first discovered in prostate gland secretions. Okay, so what is a prostaglandin? This is basically a fatty acid that's been modified. Fatty acids are what your plasma membranes are made out of. Okay, so these are released by many different types of cells into interstitial fluids, and they're going to regulate nearby cells in a variety of different ways depending on the tissue. So one, they are going to promote fever and inflammation. Okay, so they're going to intensify the sensation of pain, and so this is going to sort of contribute to the body's defenses in the immune response. We've got in semen, of course, they're going to be a component of semen. They were originally found in prostate gland secretions, right? So what do? They're going to cause the smooth muscles of the uterine wall to start to contract, draw semen into the uterus in order to cause an insemination to occur. We're going to have the placenta can secrete prostaglandins, and that can also cause uterine muscles to become more excitable and trigger the effects of uterine contraction and labor initiation and progression. They can also regulate aggression of platelets. Okay, so they can regulate how well platelets like to aggregate and cause blood clots. And because of this, like one of the treatments for high blood pressure or people that are at risk, not really high blood pressure, but people that are at risk of heart attack, right? You have maybe a lot of blockages in your coronary arteries. You have a small blood clot that could potentially cause a heart attack. How are we going to avoid that? We want to decrease the ability of the blood or the tendency of the blood platelets to aggregate. We want to basically create some way of keeping the blood thin. Okay, that is what aspirin can do, because aspirin and ibuprofen are inhibitors of the production of these prostaglandin molecules. So we have within our plasma membrane here, okay, it's membrane phospholipids. There's a lot of arachidonic acid, okay, or that gets converted here, and then these COX enzymes can convert it to this protein or this sort of, I guess, precursor to prostaglandins, and then in a variety of different tissues will lead to the production of different signaling molecules. Okay, so all of these different sort of things are these different sort of targets come from this originating structure here. So aspirin and ibuprofen are going to actually inhibit that production of this, and so they will reduce inflammation, right? They will reduce the ability of blood to clot. They're going to reduce the perception of pain. Okay, so these are effective pain relievers that are non-steroidal anti-inflammatory drugs and non-opioid based pain relieving drugs. So they operate by a distinct mechanism, not through direct analgesia, but through the blocking of the action of these prostaglandins or in the production of prostaglandins. Any questions? Okay. Okay, so those are two classes of things that are sort of local regulatory things, local regulatory signaling molecules. Now let's see for hormones, how are they going to operate? What's the general sort of pathway that they're going to operate in? Okay, so all of these endocrine pathway, all of these sort of signaling systems in the endocrine system really are part of pathways that are going to have a particular molecule that's released, a set of receptors in target cells that are going to get activated by that molecule. And then there's almost always going to be some response to that activation. right? The effector. And then there's generally going to be a negative feedback or positive feedback, depending on your system, back to what is controlling the production and release of those endocrine molecules. Okay, so let's look at one example. So if we look in the gut, okay, the gut has a particular level of pH that it needs to maintain, okay, in order to promote efficient digestion. Okay, so inside the gut lining, these S cells of the duodenum of the gut, sort of the inner lining of the gut, these can sense what the overall pH level is in the gut. Okay. If we have low pH in the gut, we're going to secrete secretin, okay, which is a hormone that's going to then travel throughout the body. Okay, it's going to hit our target cells in the pancreas. The pancreas will then produce bicarbonate. It's going to come down these sort of ducts from the pancreas into the gut. And what is bicarbonate going to do? That acts as a pH buffer. It's going to sort of bring the overall pH level back up towards the sort of buffering zone of bicarbonate. Okay, so that's going to raise the overall pH in the gut. And then that raised pH in the gut will sort of then reduce the stimulus of the low pH on the endocrine cells. And we're then going to reduce the release of secretin. Okay, so this is how we're going to have an example of negative feedback mechanisms maintaining homeostasis within the gut pH. One of dozens of examples. Now, what about, so I showed you a negative feedback. Okay, most of the examples are going to be negative feedback. What about a positive feedback? Does that ever happen? Of course. It happens oftentimes in mating and childbirth related things. Okay, so we have anything with to do with sexual reproduction, oftentimes it's going to have a positive feedback associated with. So here's an example, oxytocin signaling. Okay, oxytocin does a number of things. And one of the things it does is it is involved in the milk letdown reflex. So we have some sort of stimulus here. Okay, we have the baby's mouth on a nipple. It's going to cause mechanotransduction and our sensory neurons in the skin. It's going to then send electrical signals to the hypothalamus and then into the posterior pituitary gland, where there's these neurosecretory cells, which I mentioned are going to release neurohormones. In this case, it's called oxytocin. That hormone is going to get released into the blood. It's going to travel around the blood. It's going to hit mammary glands and the smooth muscle of a mother's breast shortly after childbirth, and that is going to cause the relaxation of those smooth muscles and release of milk that's been built up into the baby's mouth. Guess what that's going to do? That's going to provide positive feedback, right? Milk hits the lips, lips are going to start to suckle more, right? And so we have this sort of example here, right, where we have a positive feedback loop where the stimulation of the nipple is going to continue to cause the baby to suckle and cause more and more oxytocin release and a greater sort of release of milk into the system until what's going to stop this in general is going to be the baby. Okay. So then you're going to stop the stimulation and then the system will wind down. Okay. Now that's not the only area where oxytocin is going to be signaling. Okay. Oxytocin is the key driver of contractions in the initiation and maintenance of labor. Okay, so childbirth, also known as parturition, is triggered by by a variety of different molecules, but one of the most immediate triggers here is stretch receptors in the bottom of the uterus. So once a baby's head is turned into a lower position and is sort of engaged against the cervix of the uterus, we're going to have pressure here that's going to cause stretch receptors to get activated and send positive feedback right back over there to the hypothalamus, posterior pituitary gland. It's going to cause the release of oxytocin. And if the body is in the appropriate condition here, that is not going to cause a milk letdown reflex. There's no milk yet that's produced until a postnatal period. Okay, but what it will do, it'll start the uterus to start to contract and squeeze the baby and that what will the squeezing the baby do here? It's going to increase the pressure of the head on the uterus or on the cervix, increase pressure on the stretch receptors, and provide a positive feedback loop here. And then progress labor. Now you can actually drive if you know your doctor, if if the pregnancy's gone on too long, maybe you're 41 weeks in, okay, and the doctor's concerned about maybe maybe getting too big, or maybe the doctor just wants to have a delivery on a weekday, not a weekend, doesn't want to come in. You can induce labor through the injection of Pitocin, which is just the brand name version of oxytocin that will trigger that onset of this positive feedback. Okay. Let's see here. What else do we want to cover here? Okay, let's look at the pancreas. Well, I guess one thing that we should also say about sort of this set of, we want to know the anatomy, we want to know the source tissue, we want to know the target, we want to know the effects. Another thing that we want to know is what's going to happen, what will go wrong, what diseases are caused by if we have too much or too little of this substance that's being released. Okay. So sometimes you can have immune responses, like an immune system attacking a particular tissue and destroying that source tissue could result in too little of a hormone being produced. Okay. On the other hand, we could have things like adenomas, which are cancers of the adrenal cortex, which are going to cause production of cells that produce too much of that hormone. And now we have an elevation of that hormone level within the blood, and then cause some deleterious impact on your body systems. Okay. That's just a sort of general way that those things can happen. So now let's turn to the pancreas and its role in blood glucose level regulation as an example of sort of source, target, function, and disease. Okay, so the pancreas is an organ that is instrumental for digestion and coordination of the digestion products being taken up into the bloodstream and into the body cells. Okay, so it sits here, that it's sort of surrounded by the small intestine. Okay, it has bile ducts within it that can excrete digestive enzymes, can just excrete bicarbonate to regulate digestion and pH levels within the gut. Okay, but it also, so that's what most of the pancreas does. Okay, so it has basically digestive exocrine function. Okay, so exocrine means you are releasing something that then has a direct effect, whereas endocrine is where you are releasing something that then causes another cell in the body to have an outcome. So the exocrine stuff is going to actually just directly digest stuff. Okay, now, so most of the pancreas is doing this exocrine function, releasing digestive enzymes, bicarbonate. Then we also have this hormone secreting portion. About 1% to 2% of the mass of the pancreas is devoted to endocrine signaling through the islet of Langerhans, or islets of Langerhans, the little islands of Langerhans. Langerhans, the person who discovered these for microscopy, this is what they look like. So embedded little sets of cells within the vast array of the rest of the pancreas. Okay. Now, these pancreatic islets contain beta cells and alpha cells, which are going to release two different hormones that oppose each other's function. Okay. The beta cells make insulin, the alpha cells make glucagon. Okay. When your glucose is gone in your blood, you want to make glucagon. When your glucose is too high, you're going to want to make insulin. Okay. And the pancreas was sort of discovered to be vitally important to the disease of diabetes mellitus around a little bit before the turn of the previous century in the late 1800s. Okay? And this was discovered because if you removed, as the doctors and scientists were trying to figure out what is the origin, what is the cause of diabetes, they removed the pancreas from dogs, and they discovered that you were able to induce diabetes within a dog. Okay. So what is diabetes mellitus? Diabetes mellitus is an inability for you to take up blood glucose into your body's tissues. Okay. And So that will then lead to, in the case of where you have no insulin, and that's the case, you're gonna basically be wasting away, okay, as you grow up. If you genetically have a genetic disorder that does not allow you to create insulin, then even though you're consuming, food, consuming glucose is going up in your blood, you can't take that glucose in, okay? So that is going to cause your body's metabolism to rely exclusively on fat and muscle protein metabolism. And so you're not going to be able to grow. You'll be very thin. Eventually, you'll die, okay, at a very young age. So there's a pretty serious disease. Okay. And Frederick Banting, his assistant Charles Best, tried to investigate, how could we, if the pancreas is involved in this, there must be something in the pancreas that could potentially counteract this disease. Okay, so what they did is the first thing they tried to do is, okay, so there's got to be something in the pancreas that's being produced that is causing, that is preventing this disease. They took the pancreas out of the dogs, they ground them up, they tried to extract out different sort of fractionates of that, inject them into dogs that didn't have the pancreas and see what they could sort of cure, and nothing was working, okay? And then they sort of realized, okay, well, there's these little islets of Langerhans in there, and most of what the pancreas is doing is digesting stuff. Maybe the problem is we're taking the pancreas out, okay, and we're chopping it up in a blender or whatever, okay, and whatever the digestive enzymes that are produced by the pancreas, maybe those enzymes are chopping up the molecules that we need to signal in the system to cure diabetes, okay? So the next step and the key insight that they had is that they then ligated the veins or the arteries that were feeding into the pancreas, okay? And they ligated, I think, the ducts of the pancreas, and that caused a specific death of all the exocrine tissues, but it spared the islets of Langerhans. So all these other tissues sort of died off, and then they extracted those pancreases out, ground them up, injected them into diabetic dogs. Immediately the dogs got it. Okay. Then they took the same extract, they injected in a 14-year-old kid who was wasting away of diabetes mellitus. Instantly he was better. Okay. Complete change, complete sort of revolution in sort of treatments of disease was through the extraction of insulin. Okay, now they did patent this, insulin discovery. Okay, they licensed it to the University of Toronto for $1 because they really believe that it doesn't belong to them; it belongs to the entire world. This is what Banting just once said, and then there was a process of discovering how we could make insulin recombinantly. So we take the DNA for insulin, they were able to produce it in cell cultures and produce vast amounts of insulin instead of having to basically take pigs or dogs and ligate their pancreas and digest them, which is pretty gruesome. We were able to now do this in cell cultures, create that. And then, so the price of insulin, generic insulin, was cheap for many, many years. And then, of course, capitalism took over and there was consolidation within the healthcare industry, and they figured out ways to charge excessive amounts for insulin. So now insulin's expensive again. So, but that's a sad story. But the happy story is that this basically, the delivery of insulin basically cures the appropriate delivery of insulin cures type 1 diabetes. Okay, and why does it do that? It's because insulin is the key signaling molecule that allows glucose to be taken up from the blood into cells. Okay, so the 14-year-old, right, he had type 1 diabetes. He had very high blood glucose levels all the time, but he wasn't able to turn that glucose into his body, into like structures within his body. Okay. So insulin is an anabolic hormone. It is an anabolic hormone of the body. So any metabolic pathway that is going to construct molecules from smaller units that requires energy is an anabolic pathway. This is the opposite of catabolism, which is the breaking down, where ATP, breaking down of structures to produce ATP. So ATP is going to sort of catalyze anabolism. It's going to promote the absorption of carbohydrates, especially glucose, from the blood into liver, into fat, into muscles, skeletal muscles. And we can also take glucose, right, and turn it into glycogen through the synthesis here. So this is called the glycogenesis process. We take glucose, turn it into glycogen. We have all these glycogen chains that's now going to be stored in the liver as well as our skeletal muscles. Okay, so the beta cells are going to sense blood glucose levels when they go up. They release insulin. Insulin causes, sort of promotes this glycogenesis and the extraction of glucose from the blood. Now, of course, if we have something that can bring down the glucose levels, we're going to want to have another system, another control system that can raise blood glucose levels. And this is glucagon, which is generated from the alpha cells on those islets in the pancreas. So this is a catabolic molecule. What is it going to do? It's going to raise blood glucose levels by stimulating glycogenolysis. Okay, so this is the cutting of glycogen. Okay, so it's going to chop up glycogen into these phosphorylated glucoses. Okay, it is also going to promote gluconeogenesis. Okay, so the formation of neogenesis, construction of glucose from other molecules like fats, like proteins. Okay, the liver is full of insane variety of different enzymes, right, that can catalyze the conversion of one sort of molecule into another. And so glucagon is going to sort of amp up that production of glucose in the liver through gluconeogenesis. Okay. That is then going to release that into the bloodstream, that production into the bloodstream, raising up blood glucose levels. So we've got two classes now of diabetes mellitus. Okay, so back when insulin was discovered, almost everybody that had diabetes was type 1. These days, most people, 90% of the people that have diabetes is type 2. Yes, I have a question.
Yeah, I was wondering, glycogen exists in all cells or mostly in
It's mostly in liver and muscle. Okay, so there's certain areas like yes, liver and muscles are the primary storage places of glycogen. Certain tissues have a very hard time storing glucose and glycogen. One of those tissues is the brain. That's why the brain is acutely sensitive to blood glucose levels, because they have an inability to regulate glucose levels within the cells themselves because they don't have this reserve. So that's one of the main reasons why you have to keep blood glucose levels above a particular level. Otherwise, you'll have a coma, die, because you will not have enough energy substrate within the brain. So you go below like 20 units of, what is it, deciliter, I don't know, milligrams per deciliter or something in your blood, you can go into a coma. Okay. Why would high blood glucose levels be bad? Okay. They're bad because they can impact the vasculature of the blood. If you have a bunch of sugars floating around, those sugars are going to get inappropriately attached to various proteins. So you're going to interfere with their function. You're going to get this hyperglycation of proteins, and that is going to cause like hardening of the vascular system, and that's where you see a lot of the damage with diabetes mellitus is through sort of basically vascular. Okay, so we've got two different classes of diabetes mellitus. We have type 1. This is juvenile onset, also called insulin-dependent diabetes. This is generally a genetic disorder where some aspect, or it could be a genetic disorder, or it could be an autoimmune disorder, okay, where the beta cells cannot produce insulin, okay, or the beta cells are killed maybe by an immune response against the beta cell. Okay, so we have an inability to produce insulin. If we can't produce insulin, we can't take insulin up into our cells. On the other hand, we've got type 2 diabetes mellitus, the dominant form in the world right now, or at least the dominant form in America. Okay, and this is going to be where there is still the ability to produce insulin, but the action of insulin is no longer effective. It's not able to bring glucose up into the blood. Okay, so let's see here. We've got, so our type 1, this is oftentimes an autoimmune disorder. So basically, insulin producing cells are destroyed, usually due to immune attack. So how can we treat this? Okay, well, you get insulin injections several times a day, or now I think there's sort of continuous glucose monitoring patches that you can wear, right? And pumps that can inject glucose to sort of keep your blood glucose in a nice range. So a huge amount of money in this industry, like because there's so many people that have diabetes, okay? And maybe one day we'll have a cure for it through stem cell research that can sort of take the cells within your own body, turn them into beta cells, and then implant them into the appropriate way. That's, of course, that has to be derived from your own body's tissues. Otherwise, they're not going to express the right MHC class 1s, and you're going to have an immune response against them. So type 2 diabetes is non-insulin dependent. This is how that basically progresses. Okay, so normally our stomach is going to convert food into glucose. The glucose is going to go into the bloodstream. Insulin will be released. Insulin is going to tell the body's tissues to start taking up glucose. How does it do that? It causes it acts on a variety of second messengers within a large portion of these body tissues to upregulate this glucose transporter. The expression of glucose transporters on the surface. Those glucose transporters will bring glucose into the body tissues and drive increased metabolism within those body's tissues. Okay, well, if you have chronically high blood sugar levels, okay, the tissues within the body start down-regulating the systems in which the insulin is going, the intracellular systems by which that insulin acts. Okay, so they might turn down the expression of the insulin receptors, or they might turn down the second messenger systems that the insulin receptor that normally activates. Okay, so that is going to cause an increase in beta cell insulin production because the insulin is not as effective. Okay, but the blood glucose is still high. We have these feedback mechanisms that they're telling the beta cells produce more insulin. Okay, what will happen? Okay, those beta cells are going to burn out. Okay, they're going to be overactivated. Eventually, they could have metabolic diseases within the cells themselves or die out, and that is then going to cause a sort of catastrophic failure, a catastrophic drop in the body's ability to produce insulin. At that point, you're going to have to get insulin yourself, right? You've lost the ability. So what are the treatments for this? This is typically because this is typically associated with diet and exercise, right? So the more weight that you put on, or the more sugary foods that you consume, and maintain sugary foods, maintain a diet of sugary foods or very high calorically high foods, that is going to tend to lead to development of type 2 diabetes. I mean, the incidence has increased like tenfold over the last generation here. So first, the first defense against that is change in diet and exercise. Now we have those GLP-1 agonists that can actually really help in shifting the behavior of the drive for caloric consumption, as well as somehow these other like interesting knock-on effects on drives for risk as well. And then sort of the third line of defense is injection of recombinant insulin once you reach this insulin resistance stage. Okay, I guess just to sort of highlight the importance of this, we've got at USC themselves, there's a huge group of research by people like Ray Stevens, Vadim Ferzov, which are people that were recruited to USC, and their one of their major projects is to create a complete computational model of the beta cell to fully understand how the beta cell signaling works and how it will produce insulin, and how you can potentially reprogram cells to create beta cells that will produce insulin and cure, basically a long-lasting cure for diabetes. And I think that's a great place to stop for today. So thanks everybody for your attention, and I'll be on a Q&A in 1 hour from now. to answer any of your questions about exam 2 or this content, or exam 3 or this lecture. Hey, how's it going? So I'm gonna be, there's a bunch of people coming in today, after the Q&A. So, Q&A is, gonna be 11:45 to 12:45, so you can swing by this early afternoon today would be good, if you have time today, or do you wanna do it?
Yeah, you can push them around like.
Okay, come by it too.
Okay,
cool.
Yeah, I just wanted to ask like about, just like it's so I do want to do better on this.
Yeah. Yeah.
Just because like I'm actually I skipped over a bit and like if I'm having a little bit of weeks, it's like having slides. I don't even want to do and all of that, but I just want to figure out like watch.
Yeah, I mean, this study, the lectures are the most important,